Hi everyone, Dr. Mike here. In this video, we're taking a look at carbohydrate digestion and absorption. We're moving our way through the gastrointestinal tract and having a look at where and how carbohydrates are digested into their monomers, which we call glucose, galactose, and fructose, and where they're absorbed. Let's take a look. Okay, to begin, let's orientate ourselves with what I've drawn up on the whiteboard. We have our teeth and our oral cavity. I know that's frightening, but just go along with me. Oral cavity, we've got our salivary glands, and this one's going to represent an important one called the parotid gland. We have our esophagus moving down into the stomach, then from the stomach into the duodenum, which is the first part of the small intestines, and then we've got the rest of the jejunum moving through to the ileum. Here we have a duct. This duct, not quack quack, but a hollow tube, is connected to both the pancreas and the liver gallbladder, and they squirt their substances into the duodenum. That is important for digestion. Here we have a cell of the small intestines called an enterocyte, and then we've got our bloodstream here, which is representative of the portal system, the portal vein that's traveling ultimately to the liver. So we need to understand the absorptive process here. But to begin, we need to talk about ingesting carbohydrates. So there's three major carbohydrates that we ingest. We ingest starch, we ingest glycogen, and we ingest cellulose. These are the three major types of carbohydrates that we take in. Now, importantly, you need to understand that when we look at carbohydrates, the term that we use is polysaccharides, and that means many sugars. Polysaccharides tend to get broken down into oligosaccharides, which means, so polysaccharides is a very long chain, it's many. Oligosaccharides means two or more, but they're not as long as the polysaccharides. And then we need to break that down into what we call our monosaccharides. And the monosaccharides is what we absorb. That's really important. We cannot absorb polysaccharides. We cannot even absorb the oligosaccharides or even some disaccharides, which are two sugars. We must absorb the monosaccharides. So just keep in mind that that's what we're aiming for. We need to absorb the monosaccharides. All right, so the polysaccharides that we're taking in, starch, glycogen, and cellulose. Let's have a look at them. Starch, we take these from plant material. So this is a plant-based carbohydrate. In actual fact, so is cellulose, but there's a big difference. Actually, it's a minor difference, but an important difference between the two. Glycogen is animal carbohydrate. Now, every time I say this to my students, they go, I didn't know we got carbohydrates from flesh, from meat. Well, we do. Remember that our skeletal muscle contains glucose in the form of glycogen, that's stored glucose. So animals are no different. And skeletal muscle is one of the major sites of glucose storage. So yes, we can take in carbohydrates in the form of glycogen from animal flesh. Now, if I were to draw up both starch and glycogen, they look quite similar. They look like this. So remember, a basic glucose or fructose or galactose sugar molecule is a 6-carbon molecule. So effectively it looks like this. It's a 6-carbon ring. And what we have is an oxygen there, a carbon, a carbon, a carbon, a carbon, a carbon, and a carbon. And if we were to count them, we have our first carbon, our second carbon, our third carbon, our fourth carbon, our fifth carbon, and our sixth carbon. So that is the typical 6-carbon ring of a glucose, galactose, fructose molecule. And all three of them are C6H12O6, which means there's 6 carbons, which we've drawn there, 12 hydrogens and 6 oxygens, which I haven't drawn up. But just keep that in mind. So again, if I were to draw up the starch and the glycogen, they are stretches of these 6-carbon rings with branches coming off them. Now I can't draw the whole thing up, but I'm going to draw a couple for you. So starch and glycogen are very, very similar. they look a bit like this. So you can see that they're bonded together like that, and they have some branching as well. So this is what starch and glycogen look like. There's multiple branches, very long molecules. You can see the bonds in between. Now, if I were to label those bonds, let's have a look, right? I said that that's the first carbon there. So that means that that is a first carbon. And that's a fourth carbon. So that there is a fourth carbon. 1 to 4. So what we have for both starch and glycogen are these monomers, these monosaccharides. They're bound together by what we call an alpha 1 to 4 glycosidic bond. An alpha 1 to 4 glycosidic bond. Really important, keep that in mind. Now, if I were to draw up a cellulose sugar polymer or polysaccharide, it would look like this, really similar. Okay, that's looking pretty much the same at the moment, but then what you see is the bonds start to slightly change. See how this bond here is the alpha 1 to 4 glycosidic bond. That's an alpha 4 1 to glycosidic bond, but this one, it's in a different configuration. It's still what we call a, got that mixed up, a 1 to 4 glycosidic bond, but it is what we call a beta 1 to 4 glycosidic bond. And you might be thinking, oh my God, I don't care why you're telling me this. I'll tell you exactly why. Because when we have these carbohydrates in our oral cavity and it stimulates our salivary glands, particularly the parotid gland, now parotid means near the ear, it's going to trigger this salivary gland to release enzymes. One important enzyme is called alpha amylase. And what alpha amylase does, remember, these are molecular scissors. They chop, chop, chop. What do they chop? They chop alpha 1 to 4 glycosidic bonds. So it starts to chop the alpha 4 glycosidic bonds. But we have a beta 1 to 4 glycosidic bond for cellulose. We do not produce the enzyme, we do not produce a beta amylase. What that means is we cannot digest cellulose. And cellulose comes from certain plant materials. So we get this in grasses, which we don't eat, but we can also find cellulose in lettuce and the various indigestible fibers. So what that means is cellulose will move through our entire digestive tract and it goes undigested. And that's what we call fiber. So we're not using cellulose to absorb or to make energy, important. But we do use starch and glycogen because they contain the alpha 1 to 4 glycosidic bonds. Importantly, the alpha amylase breaks the 1 to 4 glycosidic bonds, but not the terminal one. So it can't break the one in the end. That's important. And remember, they're big, long chains. So this is what's happening in the oral cavity. What will happen now is that this, which is still a polysaccharide, will move through the esophagus, and it will move through the stomach. Obviously the stomach churns and everything, but it will move through the stomach relatively unchanged. Now I'm going to draw this up without the hexagonal six-sided shape because it's just going to be a lot easier. And so what we have is we're still going to have quite a long molecule that is still branched, right? Now I didn't tell you about what that particular bond is. Let's write this down, it's important. This bond, as you can see, that's a 6 carbon, so this is going to be 6, and that's number 1 because it's on its side. So this is what we call an alpha 1 to 6 glycosidic bond. That's an alpha 1 to 6 glycosidic bond. Alpha amylase cannot break down alpha 1 to 6 glycosidic bonds, so it remains attached to this polymer. All right, as we move through the stomach into the duodenum, first part of the small intestines, All these substances, remember, because you very rarely ingest carbs in isolation. You'll often ingest them with fats and proteins. And the fats, the proteins, and the acid in the stomach trigger some cells. These particular cells line the duodenum, and these cells are called they're called entero, which is referring to gut, enteroendocrine cells. Now, what does endocrine mean? Endocrine means it's going to release a chemical into the bloodstream. And the chemical that it releases into the bloodstream is what we call CCK. Now, CCK stands for cholecystokinin. Cholecysto means gallbladder, kinin means contract. This chemical tells the gallbladder to contract. Now, this is not an important function of CCK in the context of carbohydrate digestion. But that's okay, we'll talk about it in the fat digestion video. But what CCK can do is it can travel to the pancreas. And CCK triggers the pancreas to release its pancreatic juices. So it's going to trigger the pancreas to release its pancreatic juices. Now, these are enzymes. And one of the important enzymes that it releases is alpha amylase. And you might think, we've already released alpha amylase. We released it in the oral cavity. This is virtually identical alpha amylase, virtually, not exactly. But what you need to know is this alpha amylase is far more potent, which means it's going to further digest the alpha 1 to 4 glycosidic bonds. These ones far, far better. Now, once this alpha amylase has acted upon these polymers, what we're left with after this entire process are some disaccharides and trisaccharides. So 2 molecules of sugar snap together and 3 molecules of sugar snap together. Let's draw these up, right? So this is what we're left with. We are left with this. A molecule that looks like that, one that looks like that, and then a couple more disaccharides. Oops, I said disaccharides. Got to pay attention to myself. And we haven't digested that alpha 1 to 6 bond yet. So these are the major disaccharides and trisaccharides we have remaining once we are in the small intestines. What are they called? This first one here is called maltose. This next one is called maltotriose, because there's 3 maltotriose. This next one is called sucrose. This one is called lactose. And I'm sure you've heard of some of these terms before. And this branch of the alpha 1 to 6 bond is called alpha dextrin. This is what we're left with once we are past the duodenum into the jejunum, right? All right, so you've heard of maltose and sucrose and lactose before. These are disaccharides. Very important. One important point here is this. We're about to talk about the enzymes that can break these down. Some people don't contain one or more of these enzymes to break these down, which means some of these disaccharides may remain all throughout the tract. And you cannot absorb these disaccharides. They must be the monomers. They must be the monosaccharide. And that means that as it moves through, it starts to pull water towards it because sugar molecules, like glucose, have a negative charge associated with it. And it pulls water towards it if it remains in bulk in the bowel. And you get diarrhea. So people who are lactose intolerant, they don't produce lactase, the enzyme, that remains in the bowel the whole way through, doesn't get absorbed, and pulls water towards it, and that's why people can get diarrhea. But people can have a sucrose intolerance and a maltose intolerance. How does this come about? Because of these brush border cells that line the small intestines. So as we move through the small intestines, you get these fingertip-like projections with even smaller fingertip-like projections that we call brush border cells, and they produce brush border enzymes. And these enzymes break down these sugar molecules. What are the enzymes? Well, the enzymes that they release include maltase to break down maltose. Maltase also breaks down maltotriose. Perfect. Sucrase breaks down sucrose. Lactase breaks down lactose. And alpha-dextrinase breaks down alpha-dextrin. Sometimes it's also referred to as isomaltase. So sometimes the enzyme is also referred to as isomaltase. So if you see that in your textbook, know that it's the same thing as alpha-dextrinase. It breaks the alpha 1-to-6 glycosidic bond. So once these enzymes have been released from the brush border cells, we are left with the monomers. And there's only 3 monomers you need to know. So both maltose and maltotriose, what they release, they're just simply made up of glucose molecules. That's it. Maltose is 2 glucose molecules, maltotriose is 3 glucose molecules. Sucrose is made up of fructose and glucose. So 1 fructose, 1 glucose. Lactose is made up of galactose and glucose. So they're all made up of glucose. That's an important point. And then alpha-dextrinase is simply just made up of glucose. So what are the 3? What are the 3 different monosaccharides? It's glucose, fructose, and galactose. Now the question is, we've now got the monomer. I'm going to rewrite them. Now that I've already written them there, because we need to talk about their absorption from the lumen into the enterocyte, from the enterocyte into the bloodstream. So let's write it like this. We have fructose, glucose, and galactose. And we need to get them through. First point you need to understand is this. We have pretty much embedded in the membranes of all of the cells of the body is my favorite enzyme, and it is an enzyme, and people say it's not, but it is, which is called the sodium potassium ATPase pump. And what this pump does is it uses ATP and it throws sodium from inside the cell to outside the cell, and it actually throws 3 sodium out, and it throws 2 potassium in. Now the reason why this is important in this instance is if I'm throwing all the sodium that's inside the cell out, this cell becomes deficient of sodium. And you know diffusion, things want to move down their concentration gradient. And as this is the digestive tract and you get sodium from the food that you eat, you're also going to have sodium in here. So if this is deficient, what we're going to get is, I'm just going to draw this up. What you're going to get is the sodium wants to move down its concentration gradient into that cell. That makes sense, right? Because the cell is deficient of sodium, and then that sodium gets pumped out, and then it keeps moving down. This channel is used by glucose and galactose to get into the enterocyte. And this channel is called a sodium glucose transporter, an SGLT. That is called an SGLT, a sodium glucose transporter. What does it transport? Galactose, glucose, while it piggybacks down the concentration gradient of sodium. So that's called secondary active transport. Now you might think, what about poor fructose? Fructose has to move through by itself. For whatever reason, fructose moves through by itself. And what it's going to move through is down its concentration gradient through this channel. And this is called a GLUT5 channel. GLUT5, which means glucose transporter 5. That's what it uses. Now what we have in the enterocyte is galactose, glucose, and fructose. How does it get into the bloodstream, which is where we need it to go? This one's easy. It uses another GLUT, another glucose transporter, but this glucose transporter is called GLUT2. And they simply move via diffusion. Galactose goes down its concentration gradient, glucose goes down its concentration gradient, and fructose goes down its concentration gradient. And what we now have is galactose, glucose, and fructose in our bloodstream. Beautiful. We started with a complex polysaccharide of starch or glycogen, which we broke down using salivary amylase, breaking the alpha 1 to 4 glycosidic bonds, not the terminal, but the others. That makes it slightly smaller for by the time it moves through the stomach into the small intestines at the jejunum, triggers enteroendocrine cells to release CCK. That triggers the pancreas to release its pancreatic juices, which include alpha amylase. It further chops the alpha 1 to 4 glycosidic bonds, these ones here, to produce maltose, maltotriose, sucrose, lactose, and alpha-dextrin. Once it gets into the jejunum, we've got the brush border cells of the small intestines releasing maltase, sucrase, lactase, and alpha-dextrinase, chop, 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 breaking them up into their monomers, which are galactose, glucose, and fructose. Fructose moves by itself through the GLUT5 transporter through facilitated diffusion. Galactose and glucose piggybacks on the sodium glucose transporter, with sodium going down its concentration gradient. They move with it, that's secondary active transport. Then all three, fructose, glucose, and galactose, move through a GLUT2 transporter via facilitated diffusion into the portal system. Once it's in here, this moves to the liver. And from there, the liver can decide what it wants to do. Generally speaking, it will turn galactose into glucose and fructose into glucose, and then glucose in the hepatocyte, the liver cell, will undergo either glycolysis to make ATP or to be stored as glycogen. I hope that helped. I'm Dr. Mike. Hi everyone, Dr. Mike here. If you enjoyed this video, please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovic, at DrMikeTodorovic. Speak to you soon.